And now the last part of this research track before lunch, so we're not going to lunch right now yet. The last part is a very interesting talk about the world's first hybrid neuro-AI silicon computer. Please welcome Regina Luttge and Bert de Vries.
Thank you very much, and good afternoon. I'm just waiting for I have the screen here, it's okay, but not here yet.
Yeah.
Nee, ik weet niet.
The green button.
Drukken?
No.
No, I see them working here on it. I can hold this up. Yeah, there we go. All right, so our project is called BayesBrain, the world's first hybrid Neuro-Silicon AI computer. Our goal is to learn how to program live neural tissue. By way of acknowledgement, this is our team. We are generously supported by EASY, and we have 2 PhD students, 5 faculty members across 4 departments at the TU. So it's a very multidisciplinary project. And of course, I would like to thank our team members for this work. So here, as a motivation, we just observed that 2 computers are displayed here that do about the same amount of computations. Frontier is the largest supercomputer in the world. And here's a human brain, and Frontier consumes 40 megawatts, and the brain about 10 to 20 watts. So that's about 1 million times more power efficient for a similar amount of computation. On the other hand, a regular silicon-based computer is far more programmable than the human brain. So what we try to do in this project is to make a hybrid computer, and to show that it works, we will take a simple standard task, which is a cart pole task. You have a cart, there's a pole on it, and you want to balance the pole. It's just like what you can do with a broomstick on your hand. So you have to control the pole, and you make observations. And then we have a computer, and this is a hybrid computer. And this part of the computer, well, is high programmable, high power, and this part, this is live neural tissue. We hope it's going to be, well, low power. Our goal is to take out parts that are high power consuming from the, let's say, from the silicon part, and replace it by real neural tissue. And so we will get a hybrid computer that is highly programmable and low power. Now, in order to make that work, we first have to learn how the brains work, right? Because if we want to replace that white piece by brain tissue, well, we have to learn how it works. And so then we go back to physics, because the brain, well, assumingly follows the laws of physics. So all the simple thing, if you throw a ball, what path does it take? Well, you may remember Newton's law. What is it? Force is mass times acceleration. There is in physics a higher descriptive sort of umbrella framework that says in nature energy differences of any kind are neutralized as fast as possible. This is the principle of least action. If I write that down for big things, kinetic energy minus potential energy, I can derive Newton's law. We can do the same thing for movement of small things, energy difference, and you derive quantum mechanics. You take the difference between electrical and magnetic energy, and you derive Maxwell's equations, and so forth. So physicists like to put this formula, this is a Lagrangian on a mug, and say, well, this is how the world works. This is minimized in least time. That's everything that's going on in the world. So the question is, can we derive a mechanics, a physics for movement of thoughts, for movement of beliefs, for your perception, your learning, cognition, everything? And Carl Friston is a very famous neuroscientist in London, says yes, and here is the formula. That formula is the Lagrangian, and he says, that's it. That's the only thing that's going on in the brain in terms of information processing. And he derives this from first principles, from physics. It must be true. It's all that brains do in terms of information processing. The evidence for this theory pours in from all kinds of sides, from the physics, even from mathematics, from control theory, and recently also in vitro. It's been established this is really what brains do. They minimize variational free energy. Okay, so what we have been doing over the past few years in my lab, which is the electrical engineering lab, is create a toolbox that minimizes variational free energy, just like brains do. And that toolbox that looks as a graph where there are nodes, and these nodes send messages to each other. Not surprising that it looks that way. That it's very much like a model of the brain. But we didn't set out to make something that looks like the brain. We just wanted to minimize variational free energy, and this is the best way to do it. So it's not a surprise that the brain actually does it this way. And then we programmed a computer. So our computer program is implemented with this toolbox. So our controller does only variational free energy minimization and nothing else. And this is a simulation of, basically of a card. I mean, it's still in software. Of a card holding up this beam. So this is working. It's all in software. There are also things we can improve in the toolbox. We're not the first to solve this problem. We are the first to do it solely with variational free energy minimization, because we want to build something that really works like a brain, so that we can pick out a piece and say, and now we want to train brain tissue to do that. And so what we need to do is to communicate to brain tissue, let brain tissue learn how to do that and communicate back. And then if the pole stays up, then it's working. And so we've the whole system set up now to communicate between our simulation and brain tissue. This is where we're at, right? It's not fully in life working, but we are about, this is about where we are. And yeah, then I'd like now Regina, to invite Regina to talk about brain tissue, because I know nothing about that.
Thank you, Bert. Actually, I'm still wondering how AI tricked me into this project, but here I am. So we are in the mechanical engineering department. My group, Neuro-Nanoscale Engineering, works on talking with brain cells, simply put. And Bert already introduced these multi-channel systems. That is really the connecting point between what the biology is doing and what, in the end, the algorithms may do. And so how could we actually build something like brain tissue out of the body? And people in that field, obviously, they work with Petri dishes. Sometimes they work just with slices taken from animals, like this poor rat here, and they put it into the Petri dish, and they say, well, that's brain tissue, I can study it, and that's all fine. But it's not really a reliable source of doing computing, because we will need, in the end, lots of tissue. Where should that come from, right? We don't want to sacrifice animals, so there must be another way of doing it. And we have to have this way of constructing the tissue as such that it really intimately talks with the electronic system. And one of such a device is actually a multi-electrode array or microelectrode array, basically a petri dish, small petri dish on top of a microchip. And that is something going around like 30 years now at low-density electrode arrays. And that's actually the good starting point for us, because we really have not a clue what the signals are that the brain cells produce. More electrodes would mean even more data we don't understand, right? So we take it easy. Take it easy. We use this relatively low density arrays, 120 electrodes, and we grow cells onto it. But these kind of structures, they are essentially flat substrates. The cells adhere on it and stretch out, and they're actually under mechanical stress. We don't really like that as an environment to grow these cells outside of the body. And we looked into it, and many other people in the field now more, confound talking about the chemistry and the biochemistry. They thought we need to have scaffolds, more three-dimensional scaffolds, fibers, polymer fibers that the cells can cling to, and that does work. But that's not my field of expertise, so I thought, go back to the chip technology. Can we make something also on a chip platform which keeps the three-dimensional state of the cell, the cellular body? And can we read this out? And apparently, we can read this out with, for example, a dye, a fluorescent calcium imaging dye. And that is just the very first preliminary experiment. And then we said, okay, we have to have these interaction points, right? So we really have to know and understand where our cells are in perspective to those electrodes. Can we have guidance features? And we do these guidance features, and we look at it at that point, and then we can start connecting it to something we call maybe an AI interface or a new hybrid computer. First attempts, just very quickly, mechanical engineering means we are working with mechanical features to actually trick those cells to do what we want, right? So we make features that we can capture cells, we can guide the neurites, and we tried nanogrooves, and nanogrooves do give us an opportunity to actually help on these platforms to interact at very specific points of synapses with the electrodes. And then when we would do it on a silicon platform, it does help us to integrate more advanced computing, which I don't do on the electronics part in this project, but here's the proof that we can interact with those features of the cellular structures and the artificial material. And just take a note, this is still a primary source of neuronal cells from rats, actually, but we want to go away from it. And of course, we have a paradigm shift in this. And thanks to funding, we can now really look into a new biological perspective where we culture those brain cells from actually human adult-derived cells, right? Stem cell technology. And then, of course, there's the physics now, explained by Bert very well, that we can now do this type of computing technologies. In the computer works great, but yeah, can we now couple also this hybrid interface? And I just want to point out that it's not really a new thing, because with these primary cells, with these red cells, there were a lot of interactions on microelectrode arrays and steering little vehicles, and that was an example of a UK researcher in 2008. And now taking this paradigm shift and taking stem cells, and we can grow those cultures in a dish. First attempts 2D, we call them human-induced pluripotent stem cells. And they build their networks, and we put them also into 3D environments to make them look a little bit more like in vivo structures. And when we watch them, when we look at them, we want to read out the electronics signaling in there. And it's really a beautiful network of cells being connected. And then when you look at the electrodes, what do you pick up is this type of very noisy signal. But every spike on the signal of this trace on one electrode will give some information on what is going on in the networks of neurons, right? And that brings us together in combining now this concept with the electronics going on in the in silico platform. And with that, I thank you for your attention, and come back, invite Bert to the stage.
Thank you very much. In a very short time, you made something very, at least for me, very complicated, very accessible. So my compliments for that. But I can imagine that there are still questions that you may want to ask them. Far away there.
I'm wondering about, because you stated that you're growing brain tissue outside of like, what is it, animal or human body. I'm wondering about like what the aim is. Is the aim to harness the computing power of the brain, or is the harness to feed like computing power that we have outside of the brain into the brain? So that's like kind of the first question. Yeah, maybe just stick with that question as a first question.
At this moment, there is no connection to the human being, right? So the energy efficiency comes from computing within the cellular network. And I think then Bert has to take over why that should save energy.
Yeah, I think, I mean, this is not meant, it's not meant that in a decade you have this computer on your desk, right? It's an investigation to, well, can we program neural tissue, and how come it's so, it's a million times more power efficient, what's going on there? So I think that those are the main goals of the project. And can we communicate to neural tissue?
Nice, okay. I understand that. Then the second question is kind of a follow-up question, because then I understand how like it can use less energy to kind of like compute things then, and that's like an interesting research field. But there was a comment made in that we wanted to move away from like using the brain cells of rats and mice, but then the body, like an animal body and a human body, is such a complex thing. It's hard to kind of like do research and ignore kind of like the whole microbiome and gut and whatever is in there. And I'm kind of like looking at this in the context of healthcare application and research that kind of like looks at how can we stimulate the brain to prevent diseases. Now, it's not that I necessarily enjoy that like animals are being used. I don't eat animals, but like doing research with them, I can actually see the value in them. So yeah, that's kind of where I'm at.
So,
what is the question?
yeah, if I
Yeah,
if I
yeah.
understand the
So
lead is
I don't know, like, is it like, cause why would we then? So for this, I can understand you want to move away, but do you also think that in general, for like the broader scope, like in healthcare, we should move away from that, like using animals in research?
That's the point. We
Yeah,
don't
that's
want
kind
to
of the
use
point, yeah.
any animal cells any further, because now we do have stem cell technology, but they are not ready yet to actually do the task that we want them to do. So we still have to learn from all these various different systems, and also from the animals, or even from us humans, of course.
All right. Thanks.
All right, thank you.
Are there any other questions?
One question.
One more
maybe a bit
question.
funny question. But how do you feed these cells? How can they survive over time?
Oh, you do have to give them some sugar, yes.
Yes.
So it's not a safe, sustainable system. It's not a perpetuum mobile.
Okay, so you just put sugar on them. How should I imagine that?
Well, it's medium with supplements, and
Yeah.
yeah, of course, it needs to be buffered, and that's very elaborate in biological experiments. This is really the bottleneck, and we try in our group with chip technology also to optimize those kind of pipetting actions by microfluidics.
Okay.
So that can be automated in the end.
Yes. Okay. Very interesting. Thank you very much. Please give a wonderful applause to them. Thank you.